All right. Hello there, everybody, and welcome to today's webcast on container closure integrity testing for pharma sterile barrier packaging. I'm Tim Wright, editor of Contract Pharma, and our webcast today is being sponsored by Nelson Labs. They're a global provider of microbiological and analytical lab testing services for medtech and pharma companies. You can visit them online at nelsonlabs.com. And our speaker today is Jennifer Gigi, an expert technical consultant at Nelson Labs. Jennifer has over 29 years of medical device and pharma laboratory experience. She has worked in the microbiology, bioburden, IDs, packaging, and validation sections at Nelson Labs, and has gained experience with a wide variety of test methods and equipment. Jennifer spent 15 years working in the packaging lab and is one of the original packaging group members at Nelson Labs. She was heavily involved in validating all the packaging test methods and equipment and writing the test procedures for the packaging tests performed at Nelson Labs. Over the next 45 minutes or so, Jennifer is going to review all the common CCIT methods found in USP 1207 to compare the testing options, and she'll also explore CCIT method validation approaches and pitfalls to enhance understanding of how methods can be applied to different sample types. We will be accepting your questions throughout the talk. You can send those in using the box at the bottom of your screen, and questions will be answered after the presentation in a brief Q&A. If you're having any technical difficulties, you can type those into the question box as well, and someone on the back end here will help you out. At this point, I'll turn it over to you, Jennifer.
Thanks, everyone. Hope we have a good time today talking about container closure integrity in testing, or CCI or CCIT. CCIT methods are going to fall into two categories, either probabilistic or deterministic, and we'll kind of go through what those mean and how they differ, and then look at all those different methods that are offered in USP 1207 to kind of get a background on how they all work and an understanding of that, and then finally finish up with method validation and explore how you would go about validating the CCI method for use. Again, as said before, we're starting with USP 1207. That is the package integrity evaluation of sterile products, and this really is just how we test pharmaceutical packaging. And this is container closure integrity, things like vials, bottles, syringes, auto injectors, and such, generally rigid containers. The probabilistic versus deterministic, to get started with that. Probabilistic methods they're qualitative. They're pass or fail, whereas the deterministic methods are quantitative. So you're getting a numerical result. And that's part of what makes these better. And if we kind of do a compare and contrast, here you'll see that these are mirror opposites of each other. When a probabilistic method is going to be generally destructive, oftentimes there's some manipulation involved. Deterministic methods are the opposite. They're going to be non-destructive. They're usually not going to require any manipulation. A probabilistic method is going to have probably a longer test time. Opposite of that, deterministic methods are going to have a faster test time. These items are going to make it so probabilistic methods are really hard for an inline manufacturing, right? You can't test the product and then, destroy it in the process. But the deterministic methods, being non-destructive and fast, are really well suited for inline inspection. And you can even set these up for 100% inline inspection, which is really the direction that a lot of this is going. Probabilistic methods tend to be less sensitive. Oftentimes that's just the way that they're set up. Where deterministic methods are going to be much more sensitive, and that's because instead of using humans to make those pass-fail judgments, we're using more costly instruments to measure a numeric value that we're comparing to our validation to get really precise data. And that's kind of the only place that probabilistic methods seem to be a little bit better on the outset, and that's because they're cheaper to set up. I can probably do some of these for 10 grand or less in equipment costs, whereas those deterministic methods, because they're using these really sensitive detectors, they're generally maybe 100,000 or more to buy the equipment needed to run one of these methods. And then the last thing is that with a probabilistic method, you really have no way in a pass-fail type of test to have any idea how to quantitate maybe leak size. Whereas deterministic methods, because I'm getting a measurement, sometimes it is possible to tell roughly what your leak size is. So in all ways, we really do prefer those more sensitive, those faster, those better deterministic methods. This is the table from USP 1207, and this is really the methods that they list. This isn't an all-inclusive list, so they've got that little caveat there that alternate methods are allowed if they're properly validated. And that's just because we never know what's coming up on the horizon. Somebody might have a great idea for container closure integrity, and we wouldn't want to exclude it until we could update the standard. So we'll be going through these methods here in the order they are listed in the standard, and that's really to kind of make sure that I don't, have any kind of preference or bias in the discussion. So as I look at probabilistic and deterministic methods, I've created some visuals here to kind of show you how these work. Most probabilistic methods are going to start with an indicator of some sort. I'm going to create a pressure differential that's going to move that indicator across the barrier, and then I'm going to end with some kind of a visual examination where I'm looking for that indicator to decide whether or not it made it past my barrier. If this is a bubble test, my indicator are bubbles that I can see visually bubbles coming through the fluid. If it's a dye test, I'm looking for that dye color to transfer from the outside to the inside of the containers. If it's some kind of a microbial test, I'm looking for evidence of microbial growth visually in my examination. When I look at the deterministic methods, they do fall into two different categories, and this option one I call the emitter detector version, and this really applies to electrical conductivity and to the laser headspace. And in this one, I've got an emitter, and if it's conductivity, the emitter is sending out an electrical signal. If it's the laser headspace, then it's a laser signal that's coming out. It's going to go through the sample and then be picked up by a detector. And the detector is going to interpret that signal and how it's changed as it passed through the sample, and then use that to determine whether or not there is a leak. In the option two, I call this one the vacuum-based option. And so these methods like mass extraction or pressure vacuum decay, and even the tracer gas when you're doing it in the vacuum mode, are going to fall into this category here. And for this vacuum-based method, I'm going to take my sample, then I have to place it into some kind of a fitted chamber, some kind of a fitted vacuum chamber, pull that vacuum, creating a pressure differential, and then I have to measure something with the detector. If this is pressure vacuum, or this is going to be, my detector is measuring volumetric flow. If it's mass extraction, my detector is measuring mass flow. If it's tracer gas, then my detector is measuring that tracer gas concentration. So let's go through these methods in a little bit more detail and kind of look at how they run and see how they compare to those general diagrams. In a bubble emission, this test is very common. We use it all the time for flexible barrier packaging. Not so much necessarily for the rigid containers, but it's a very, very valuable method to have in this arsenal because there are some container types that aren't those simple vials and syringes that we're seeing on the market today. I'm thinking about tubing sets or those big mixing sets where we have a lot of vented closures and one-way valves and such, and it makes container closure integrity in the traditional sense a little more difficult. And so bubble does still work for some of those. In this method here, the top picture, this is an ASTM F26 representation, and that's the method that we do at Nelson. On the bottom picture, I have some equipment that one would use for ASTM F3078. This is not a method that we do at Nelson currently. But when we're doing a bubble test, we're going to put it underwater. In the top one, I'm inserting a probe and I'm adding air to the inside of the package. But in the bottom one, I'm drawing a vacuum in that chamber, and that's creating the pressure differential from the outside in. And then I'm just looking for bubbles. That's an indication of air escaping from the package. In the microbial challenge, which is an immersion test, I am taking my sample containers and I'm going to fill them with growth media because I'm using microbes and I need some kind of culture media that's going to turn turbid as an evidence of growth so that I can look at that visually and see that my indicator, which in this case is a bacteria, has moved across the barrier. And that can be problematic for a pharmaceutical manufacturer because they can't just take regular samples that have come off the line. They either have to stop the line and replace the filling with a growth media, or they have to manually fill these, and then the capping is not going to be representative of the actual packaging line. So a little problematic with that. We're going to take our chamber, we're going to fill it with a liquid culture of live bacteria. The bacteria that we use at Nelson is Pneumonitis diminuta, and that's a very small organism. It's been known to sometimes even pass through a 0.2 micron filter. We're going to culture that in such a way that it's going to encourage it to be as small as possible. So kind of feeding it on a very strict diet so it doesn't have a lot of nutrients to get plump and fat because we want to keep it as small as possible to get into the defects, which again is going to take a while. Maybe you spend a week or two culturing your bacteria. Then you're going to put the samples in there. You're going to pull a pressure, sometimes vacuum and pressure, sometimes just vacuum. The vessel we have is capable of both. And then you're going to take the containers out, you're going to clean them off, and you're going to incubate them for another week and then see if the inside, if the media turns turbid, and you're looking at visually. So this test, you can see, takes quite a bit of time and expertise to perform. Then there's this tracer gas sniffer, and again, this one has a couple options. So in the top option here, the tracer gas is inside the container, and I'm just taking a sniffer probe attached to a detector, moving it manually across all the surfaces and trying to see if I see an increase in gas concentration, indicating that there's a leak and that gas is coming out of the container. This option is kind of nice because I can sometimes get a rough idea of where a leak is. But again, very technique dependent based on how far away the detector is, or the sniffer probe is, and how quickly it's moved across the surfaces and how accurately all the surfaces are covered. With the bottom option here, I've got my unit hooked up to the detector directly, and then I'm just kind of spraying it with that tracer gas. Same issues with technique. I need to make sure that I'm very consistent in covering the surface area, and that my distance from the sample is good in order to pick up leaks. So this is kind of a caveat because I'm not looking at a visual indicator. I do have a detector that is measuring, but these detectors tend to be a lot less sensitive than some of the ones that would be used in this high-end equipment. Then there's the tracer liquid. This is also a test that we run at Nelson Labs. We use a methylene blue-based dye, and we're going to take our samples. These can be regular samples, just right off the production line. We're going to immerse them in the dye, and then we're going to draw a vacuum, and that vacuum is going to create the pressure differential that'll move the dye in through any defects, and then they'll be inside the container. After we've run our exposure, we're going to wash the samples, get the dye off the outside, and then we're going to look at them and see if, the insides look blue. We also have an option where we use a UV-Vis instrument to scan the dye. Methylene blue has a characteristic wavelength that's 665 nanometers, and so we can scan for it. That enables us to see smaller quantities of dye in the solution than we would be able to see visually, but it doesn't change the test method sensitivity. So those are the probabilistic methods. And kind of get an idea that anytime that I'm relying on a visual examination, there's some subjectivity anytime that humans have to make measurements. So these are the beginning of the deterministic methods. I'm starting here with high voltage leak detection or electrical conductivity. And in this, this is the detector emitter. I have one side that's sending an electric signal. I have the other side that's receiving that signal, and they're looking for a change in the signal, which would indicate that you have a leak. And the way that it works is that the test container has to be non-conductive, and the material inside has to be conductive, and that way, if there's a leak, you'll see a spike in the current. I've got a couple pictures here just showing a couple different setups. One is in a vial, and in this one, this would spin around. Both of these, they're going to spin around, and so that the detector and emitter can test all the surfaces. These two examples are only testing the barrel of the syringe or the body of the vial. If you needed to test the closure, you'd have to have some kind of a different setup to make that happen. I like this option because it works well with pertinacious compounds. Sometimes if you have a leak and you're getting evaporation through a defect, you can plug that defect up. And so this one is not affected by that, whereas most of the other methods, that might be a factor for consideration. The next one in that detector emitter section is going to be laser-based headspace. And in this one, I'm sending that laser through. That laser is going to be tuned to a certain gas. Most of them are going to be oxygen, but you could get one that was nitrogen or carbon dioxide. And so I take my sample, the top box here, that's just a bench unit, and I would just insert my vial into that little hole at the top. I need to have fixturing so that it puts it so that the sample, so that the laser's going through the headspace, it can't go through the liquid. And it's just going to run through there, and then I'd be measuring oxygen concentration based on the detector interpreting that signal and determining what oxygen concentration that correlated to. If that's how it's set up, and this is how most of them are, it becomes a little problematic if you're packaging under normal atmosphere, because then the concentration of oxygen inside your container is going to be the same whether there's a leak or not, as normal. So one way to accomplish this is to just take the samples, put them in a vacuum vessel, add some nitrogen, create that pressure differential. That'll move nitrogen into any leaks. And then when you go to scan them, and you have to do this pretty soon after removing them from the chamber, when you scan them, then they should have a higher nitrogen concentration, therefore a lower oxygen, and then you would be able to detect leaks that way. The bottom picture here is an example of an in-house manufacturing line. So you can see I've got all the vials just going through an area where they're going through the detector emitter, and then past that, there'd be another section in the line that would be able to remove any samples that came with a failing result, so they could be inspected or discarded. One really great caveat for this method is that it can detect changes in cold storage. So when a vial goes into ultra cold storage, like -80, right, you're going to get some shrinkage in the rubber stopper, and that could allow for some leakage to take place during storage. But then when that sample was brought out of storage, moved to room temperature, the stopper is going to expand back to its normal position and seal that leak. And therefore, when it's tested, it's going to pass. Based on this method, when the stopper is in cold storage and contracted, you'll get a change in the gas mixture, and then that becomes sealed as it comes to room temperature, and you would be able to detect that with this method. So that's a caveat for this one. And then we're moving into the vacuum-based method. And the first one of those on the list is mass extraction. This is the deterministic method option that we currently have at Nelson Labs. And you can see this is our benchtop unit we use for testing. On the top, attached to the machine, is a vial chamber, and I also do on the side of that, I have another chamber for auto injectors, and we have some other ones that aren't in the picture. And for these vacuum-based methods, I'm going to put my sample in the chamber, and then I'm going to draw a vacuum. And I have to evacuate all the air around the sample, so get all the extra air out of the chamber, get it to stabilization, and then I can measure something with my detector. With mass extraction, I'm measuring mass flow. And one of the things that I really like about this particular piece of equipment is that it has a calibrated leak orifice, which is an internal defect that I have inside the machine. And I can switch that on and off with a little switch, a little black switch on the front of the instrument. And that will give me a simulated defect for any test sample that I'm running. And I love it because it means that I'm going to have a uniform defect size. And we run one of those with every set of samples that we do, so that we have a defect flow for the positive control. Now, very similar to mass extraction is going to be your pressure decay, your vacuum decay, or leak. Sometimes it's called vacuum leak. All depends on really how it's set up. And I've got an example of the equipment here. You can see that this is a very different type of test chamber. It's going to be for an oval tray. And you would just take your tray, you would put it in that particular chamber, clamp the chamber down, and then, again, start pulling a vacuum, get it to stabilization, and then here you're going to be measuring volumetric flow. Then this one is the tracer gas. This time it's vacuum mode, which moves it from being a probabilistic to a deterministic method. Usually a tracer gas is helium. That's because helium is in relatively low concentrations in the atmosphere, and so it works really well in this type of an application. But you could use a different tracer gas as well. And again, you can set these up outside in, inside out. In this example up here, you'll notice that the big difference is the shroud. So now I put my sample in a container. That means that I have a closed system, and that's going to improve my sensitivity, because now I'm not losing random helium molecules out to the atmosphere. So it's a much more controlled space. The picture on the bottom kind of shows how this might work, where I have a gas supply. This would be my helium that's in yellow, that's being charged into the product. And the sensor is picking up what's in the test chamber, and the blue would be just normal atmospheric air. But in this particular test part, there's a leak, and so it's also picking up some of the helium. There's lots of variations you can do with this, and one really good thing about the helium leak detection is that you can actually isolate closures. In the example of a syringe, you have two closures. You have the cap on the back of where the plunger goes in, and then you have the tip where you would attach a needle. And if you fixture a certain way, you can test each of those independently, whereas the other methods, you can only test the whole container at a time. Here I've got a comparison table just to kind of look at the critical parameters of these methods together. And you'll see that all the deterministic methods, the test method sensitivity is really good. It's less than 10 microns. That's very good sensitivity for regulatory bodies, less than 10 microns. And that's going to vary on a few things that we'll talk about in the next slide. The sensitivity varies a lot for the probabilistic methods, and that's because they're all so different. But the sensitivity, you'll notice, is always higher than you'll see with the deterministic methods. As far as standards go, I have some methods with and some methods without standards in both categories. I would suspect that in the future we will see standards for electrical conductivity and laser-based gas headspace. I know that many years ago, pre-COVID, maybe 2018 even, that there was someone who volunteered to start writing a standard for the electrical conductivity. I don't know if I haven't heard anything about one being started for laser-based headspace, but I would imagine we'll see one. It's just a matter of getting a technical expert who's motivated to work with ASTM and try and get a standard. As far as time goes, when you look at the deterministic methods, they are all seconds. And again, this is going to vary exactly based on how you set everything up, but every one of these can be super, super fast compared to the probabilistic methods where your best time is going to be minutes. So again, deterministic methods are preferred over the probabilistic methods. I think we've gone through quite a bit of reasons why. And then when we're looking at those deterministic methods, trying to figure out, which one is the best, they all have great test sensitivity. And the sensitivity for your method, and the reason why I said they were less than 10, is because it's going to vary on primarily these 4 factors. So the first one is method parameters. When you're trying to create your method, you can, if you're going to be very specific to a certain sample and a certain fixture, you can create a very, very custom method, and you can get your time down, and you can probably get your sensitivity down when you do that. Working for a contract lab like Nelson, we're going to use this fixturing to test a wide variety of samples because we need to test whatever comes in our door, and we get a variety of samples. So our methods are a little bit more generic, and you'll see when we go through our method that we're looking at a method that's close to a minute long. It could definitely be smaller if we tuned it, and most of these would be maybe 10 seconds or less. You could get a method. So fixturing, custom fixtures versus generic, right? So how the fixturing comes into play because it's about the amount of free space that needs to be evacuated in a vacuum-based method. And if I have very tight fixturing, it's very custom, it's going to be much faster for me to get through those pre-steps before I do my final leak check. As far as the container goes, it's the type of closure. We're going to see methods that are going to be less sensitive and maybe a little bit longer to execute with a threaded closure versus a crimped closure. And again, that has to do with removing all of the extra air before I can reach stabilization and make my measurement. And that's because I have to pull air out of all those threads, and that's going to take a little bit more time and effort. Also, the quality of the closure. If you have a really good capping process and you make very consistent caps every time, you could probably, fine-tune that method a little bit more. Most of the time, though, we see that there's a bit more variance in the final flow values, meaning that the quality of the closure has a little bit more of a fudge factor to it, and so you need to make sure that in those cases that the method is going to probably be a little bit less sensitive and a little bit longer to accommodate for that. And then lastly is the detector. I can get a much more sensitive or less sensitive detector depending on the particular machine I have, and obviously that's going to affect sensitivity. And then just the final note that some deterministic methods may have advantages that may be useful for certain circumstances, and that may influence your choice as to decide which method might be best for you. Now, this last part here, I would like to go through how one would validate a method and what you would be looking for. And in doing so, I'm going to use mass extraction as an example, and that's obviously because I validated the mass extraction method at Nelson Laboratories. I have access to the data from that validation and can use that to kind of explain this. But the principles here are going to be the same. Obviously, any vacuum-based method is going to be very, very close to this, and even those detector-emitter methods are going to follow these same concepts. The first thing that we need to look at is how does it work? because you really need to have a good understanding of what's going on behind the scenes and how changing certain variables will affect your final result, so that you can set one of these up properly. There's this video. I don't think it's going to play, just to give you a heads up, but it's on YouTube. All you have to do is look for mass extraction. You can find this. It's put out by Pfeiffer Vacuum. They purchased ATC, which is the maker of our machine, and they're the ones who really got the mass extraction standard written and got it all set up. So let's see if I can get, all right, this is a much clearer picture from here. So I'll just explain what you would see in the video, and we'll see if the video will play. It probably won't, and we'll move on from there. So on the right corner, I have a box here. That's my vacuum chamber. Inside of it is a red box. That would be my test sample. They've got a cutaway to show you all the little blue dots inside. Those would represent, air inside your sample. And you can see that there's blue dots in the outside as well. When the test starts, I'm going to do that evacuation step. That's going to draw all the little blue dots out of the outside of the box. And they're going to go down through to this bigger blue box in kind of the center bottom of the picture. And that is the first of two detectors that mass extraction instrument has. That's my less sensitive detector. And I'm going to run my first two leak checks through that detector. Then there's going to be a period of stabilization once all that has gone through. And then my valve closes for the less sensitive detector and opens for the, they call it an IMFS, Intelligent Mass Flow Sensor. That's their super sensitive detector that they're going to use for that final leak measurement. And at that point in the video, you'll see there's a little slit on this sample, and some of these blue dots would be coming out of that, and they would be picked up through that more sensitive detector. Let's see if it plays at all. No, didn't play. All right, so go ahead. There's a link on there, or you can find it on YouTube. You can watch it. Now you'll understand what you'd be looking at there. The next thing, and this is great about all of these methods for deterministic CCI, I've got a software program that runs this test. So all my analyst needs to do is take the sample, put it into the test chamber, and press a button, and the software does the rest. So what can I move or change in the software? Well, on the right-hand side, I have this time section. I cannot change any of these 13 steps. These are fixed, and these are the steps that I'm going to go through to evacuate the chamber of all that extra air and then run my leak check at the end. So I can't change the order. I can't change the steps, but I can change the amount of time spent. And you can see from step 13 that's just a tenth of a second. I could make these really fast, and that's how you can get a method that's super quick. But if you add these all up, I think it's something about 40-something seconds or so. So it's a little under a minute, and that works for us. In the center column, you can see that I have some limits, and I can actually change those. So those are my high-low limits for pressure, and I can adjust those. I can make them tighter. I can make them wider. So here is an example of the output for the test. So this is a graph, and what the test is doing is it's real-time measuring flow versus time, and then it will graph this out for me at the end. And I can see here I have this little flat section at the front. That's my gross leak check. It's just to make sure that the lid is on, that I'm getting a good feel. If I can't start to see a vacuum, if I'm pulling, if I can't start to see a difference there, then the machine's going to just terminate because it doesn't want to saturate the detectors. So that's a safety step. And then the next, I have two peaks, a little one and then a big one. That is my large leak check. That's the second leak check that it's going to run. And that one, for this particular method, if it goes above 15, then it's going to cut off the test and just cut it off right there. And that, again, those are both kind of sample prep steps. that are going to go through that larger, less sensitive detector to just protect that super sensitive detector to make sure that, that it doesn't get saturated. Then you can see I have a long, flat area. This is my stabilization phase. And then at about 42 seconds, okay, so this method is closer to 60. At about 42 seconds, I'm going to start my my fine leak check, which is my final number. And wherever I end up at the end of this curve is the number that gets reported for my final flow. This green line here is my 2 micron defect. You can see that ends up at about 12. That's my CLO, that calibrated leak orifice. So when I run my control that I'm using as my positive control for this test, I'm going to get a final flow around 12. And then you can see this this other kind of lime green line that I have going across the graph. That's my pass-fail threshold. That's the number that I am determining by my validation that anything above that line is a failure and anything below is going to be acceptable. And for this method, that pass-fail is about 5.5. And then I have this cluster at the bottom with my darker blue. This is my non-leaking sample. These are all the good ones. And you can see that I have a lot that have stacked right up on each other and have final flow values near 0. But then I have a few in the set that have a little bit higher. Most of them are below 2. Looks like I've got 1, maybe 2 that might be just, in the 2 to 3 range. But most of them are going to be below 2. And then a 2 micron defect in this same test system is going to be about 12. So if I'm setting up a mass extraction method, and this is going to be the same for a lot of them, right? I'm kind of going through these steps or these hurdles along the way to try and get my method to the point where it's working. So the first step is that I need to get the method to run to completion. And we saw in the graph there that I have those 2 early leak checks. The test will terminate if it doesn't pass those first 2 leak checks, which means I've got to get to completion before I can even make a judgment about whether or not I have a leak. So that's my first hurdle to overcome. If my method isn't working, if I can't get it to run to completion, I can look at the timing steps, maybe changing the timing that it's spending here or there, maybe change the limits. Maybe if I'm having trouble passing the large leak check at 15, maybe I'm passing it, maybe I'm going through it at 20. I could up my value to 25 or 30, and then that would get me over so I could run to completion. I might change my fixturing so that I had more custom fixtures, I had less free space in there, things like that. So once I figured all that out, I've got the fixturing and everything set up right, the method seems to be working, I can get it to run to completion. Now I need to start looking at what those final flow values look like. And I need some uniform flow values. They don't all need to be the same. And you saw in the picture, we had a lot of them that were very near 0, but we did have a spread pretty much from 0 to 2. And those are pretty uniform flow values. And then once I've gotten that, I also need to look to see if I have a difference between leaking and non-leaking flow rates. And maybe if I have a wide spread between the good and the bad parts, I might have overlap. And then I can't really set a good pass-fail threshold because I don't have space in between where there's a definition of these are the good ones and these are the bad ones. It gets kind of gray and murky in the middle where this may or may not be good. So I need some good separation between the leaking and the non-leaking flow rates. And then lastly, I need to have controls that are in range. I'm going to be monitoring, this test with every set of samples that I run to make sure that everything's in order, and so I have valid data. And so I need controls that are going to be in range. For mass extraction, my negative control is what we call a master part. It's going to be a solid metal part that's similar in size and shape to the test sample, and I'm going to use that as my negative. Obviously, if I have a high flow value for a solid piece of metal, that means that there's something else in the test system that's causing that high flow. Maybe it's residual mass from the air in the chamber because I didn't get it evacuated out, or maybe I've got something in the test system from the previous sample that's gumming up the works. That's why I need to have I need to have that negative. And then my positive is going to be that same master part with the CLL on. So a simulated defect in the solid metal part. And I need to make sure that the flow values for my positive and negative are going to be similar to the flow values for my samples and for my samples with the CLO, so that I know that I have a good comparison, and that my controls that I'm using for testing are reflective of the right flow values for that particular setup. Now, I just wanted to kind of show you when we talk about understanding the method so that you know how things will affect your final output. Just give you a couple examples here from this particular validation to kind of illustrate how things can become different. So in this particular slide, I have a comparison of the same sample on two different methods. So I've got one set of data here and another set of data here. And the only difference between these two methods is the same sample. And by same sample, I mean the exact same sample. And it's the same fixturing. We've got the same chamber, the same fillers, and everything. It's all the same. The only difference between the methods is that the very first evacuation step is double in time for one of the methods versus the other. And you can see that does, changing that one little timing step changes my average from being about 3.2 to being, about 2.6, 2.7. So, and it's not a matter of, that they got leaky or less leaky. It's just the flow values by changing that one timing step. Here's another example where I have different samples on the same method. And these are very different because I have one set of syringes that are in a syringe fixture. I have another set of vials in a vial fixture. And both of these fixturing setups are very optimized. So I don't have a lot of free space. These are really good fixturing setups. But still, you can see a significant difference in the flow values for both the leaking and the non-leaking parts. The red line here is representative of the pass-fail for the method. Again, it's about 5.5. And you can see, anecdotally, this is how the method was run. It was run on the vial samples, and then we added the syringe samples later on. So you can see that the pass-fail is very well balanced, right in the middle of a leaking and non-leaking flow value for that particular setup. But then we wanted to use the method for the syringe fixture and see how that worked. It does work. I do have, the good separation, and I can draw that pass-fail right there. And so I can use it, but I'm going to get higher numbers, and I'm going to have a greater opportunity for having an outlier that's going to fail my test, which is fine because, it's a more conservative approach. But just to kind of see the difference, same method, just different fixturing. All right, so now that we've got a really good understanding of the method, we figured out how to get the method set up, how to optimize it, getting the right chambers, the right fixturing. You saw we have that one chamber. I actually have a setup for 8 different sizes that can go in that, and I have what's called fillers that will just fill up that extra space and make that work with, so I can use one chamber with a lot of different sample sizes. If you were using one of those detector emitter methods, you might need some adapters or holders to place the sample in exactly the right position so that you could run those tests. But let's assume that we've got all that test set up, taken care of. We figured out our method, and we've got our parameters all wonderful. We've passed all those hurdles, and we're ready to go. At this point, it's time to start testing actual samples. And what we'll want to do is we'll want to have a lot of good parts or non-leaking parts. And then we're going to collect that average result. And then we're going to need some defect parts and then collect that average result. For mass extraction, I just use the CLO. And so I have that same exact defect with the sample, and I can get a really good average value that way. If you were using another method that didn't have an internal defect, then you would need to create some kind of uniform defect parts that you could get that average result for what a defect of a known size would look like. You're going to want to do this with multiple analysts over multiple days. You may notice from our validation that we are very fond of the number 3. So our analysts have done the testing 3 times on 3 different days. You don't see that because I only plotted day 1, but yeah, we love 3. Some people also like 2, and that's fine if that's you. Once you've gotten all that data, now it's time to crunch the numbers, analyze the data, and figure out where the appropriate pass-fail value should be. So just a few thoughts about defects here. So every type of defect may have its potential issues. We see lots of people using laser drilled holes. If you're going to do that, make sure that you use a good vendor who will certify their defects. And that's because flow through a defect will change as the thickness of the material changes. And so you don't drill a 5-micron hole to get a 5-micron defect. It's going to be a larger hole. And the size of the hole is going to depend on the thickness of the material. So they'll flow test them, make sure that they are the equivalent of whatever defect size you've asked for, and give you a certificate. Another one would be microtubes. We've used those before. They're kind of like a GC column tube, and so it's got a uniform diameter inside. But as you're cutting it to make a little sample, because it's on a big reel, you could get shards inside, and you have to be careful about the length for the same reasons that I just discussed. And then our favorite, we like these microcapillaries. The picture in the bottom right, that's an example of one of these microcapillaries. They come to a point, and that helps to mitigate the problems with having the really narrow leak pathway, because it's a much larger leak pathway for the majority of it. This looks really big in this magnified picture, but these actually will slide into the inner diameter of a 16-gauge needle, and that's how we actually puncture them through, because they're extremely, extremely fragile. That's how we get them through a septum. We like these just because it's something that the lab can do. It doesn't really require any specialized equipment. And then the picture on the bottom left is an example of what our CLO for the mass extraction machine looks like. We send this out once a year back to the vendor, and they will check the flow on it and make sure that it is the same flow that it's been showing for all this time. And inside of that casing that's going to screw directly into my machine, I actually have one of those glass microcapillaries. And then, see, that covered them. Yep, that covered, nope, I missed wires in the steel. You can take a wire and you can put it under the rubber stopper. And this is a nice one because it's very easy to do. It doesn't require any specialized equipment. However, the effective leak size is going to vary. You can't just take a 2-micron wire and stick it in there and get a 2-micron leak, because the leak is actually the space around it. And so you'd have to just experiment around by putting different diameter wires in the seal, and then flow testing them, figure out what equates to the right defect size you want, and then you'll be good to go. All right, so getting ready to wrap up here, let's look at our example validation data and kind of see how we did. So if we look at our first hurdle, did the method run to completion? Yes, it did, because I have data, and so that lets me know my method ran to completion. Do I have uniform flow values? When we're looking at the non-leaking and the leaking parts together, it changes my scale. And in this view, I can see that I really do have some very good uniformity in my numbers from day to day. And these are the same set, because it's a non-destructive test. These are the same set of 30 samples. That's what we used for this particular validation, run over and over. But we will see variance from day to day, and some of that has to do with the ambient humidity that's in the air. So you want to take those types of things into account in your validation. That's why we do it over multiple days. But I have a big space between my good parts, which are somewhere around 3 in this set of data, and my leaking parts, which are just over 12. And that does correspond well to the example of a just typical sample set that we had, where those flow values were between 0 and 2 for the good parts, and the defect that we had, our positive control for that one, was about 12. And so, yep, this will work great with the method. Those are right in range, and I have plenty of space to draw my pass-fail threshold. So that covers hurdle number 2, uniform, and hurdle number 3 got a good difference. And then we looked at our controls in range and saw that our negative control for this is going to be really close to 0. And that's, we do see that sometimes that the master, because again, it's a 0 leaker, that it will be lower. And we're going to be in about the same range, though, with the CLO. And so our controls are good with this method. And then just to give you an example of what a master part would look like, this picture here at the bottom, that's a master solid aluminum, shape of a vial. Maybe not exactly a perfect match, but again, that's because our fixtures are meant to be a little bit more generic. All right, so this slide here just has my contact information. If for some reason you need to get a hold of me, and then a quick plug for the Sotera Academy. If you like the content that you've seen today, we have dozens and dozens of webinars on various topics that are available on the Academy should you want to sign up for free and peruse any of that content. And that puts us to the end here, and I see that we do have some questions in the chat and a few minutes left to try and get through some of these.
Okay, great. Yeah, thank you so much, Jennifer. Great job. It was a really informative talk. And yeah, like Jennifer said, we do have time before the top of the hour to answer some of your questions. If you did have a question for Jennifer and weren't able to get it in during the talk, now's the time you want to go ahead and type it into the question box there. And if we do run out of time and aren't able to get to your question, Jennifer will have your contact details to follow up with you directly. So, all right, let's see here. This attendee says during initial closure validation, our plan is to use media-filled vials. What deterministic method is compatible, and are any deterministic methods compatible with WFI or DPBS fills?
Okay, so these container closure integrity tests are not really dependent on what's inside. The deterministic ones, they're going to be they're just dependent on the closure being good. And so you could fill them for your validation. If you're using media-filled vials, then obviously that's more of an aseptic processing validation. You're going to run your batch of product through, fill them with the you're going to fill them with the media. You could send those directly for a bacterial immersion. But if you wanted to use the better deterministic method, it should be just fine to test them because it doesn't really matter what's inside. Works fine with water for injection or drug product vials. The only caveat is that regulatory bodies are going to be looking to see that you're testing your container closure with the drug product because they want to make sure that there isn't any kind of degradation that's happening, that your drug product isn't destroying the rubber septum or something to make that closure no longer viable.
Okay, great. Thank you. Moving on, can CCI replace sterility in the stability protocol?
Definitely. There was a document in with the FDA. It's an FDA guidance document. It's like from 2008. It's container closure integrity testing in lieu of sterility for stability or something. Should be able to find it anytime I've kind of looked up, and I just that in lieu, that really works well in the search. But the idea in that document, and again, that's way back from 2008, is that physical test methods have a preference over sterility test methods because you're faster and you don't have the microbial contamination issues. So they would prefer to see a physical test like one of these deterministic container closure integrity tests as part of the stability protocol rather than relying on sterility testing. And there's a webinar on the Sotera Health Academy that's going to talk about proper uses of sterility testing and why you wouldn't want to use sterility as part of stability. So yes, please replace sterility with CCI testing for stability.
Okay, thanks, Jennifer. Let's see here. This attendee asks, how much pressure has to be applied during aluminum seal crimping?
That's going to depend on your cap. It's going to depend on your crimp. It's going to depend on your septum and your bottle, your vial, and that's usually figured out during your process validation. So you would want to play around in your design phase with different settings. You probably get some settings from the manufacturer. You want to play around with settings to try and optimize that. There is a test that you can do called residual seal force, and you're essentially using a tensile tester to press down on it once it's been capped, and they're looking at the pushback to see if they get a lot of pushback. That means that you haven't sealed it very good, so you'll have a high residual seal force. If it's crimped down really tightly, then that rubber septum is compressed and it becomes more rigid, and then they're not getting the same kind of pushback. So that's kind of the method that I would use to figure out if you're using the right settings for your capping. And then container closure really doesn't measure how strongly it's capped. It's just measuring whether it's capped enough that it's not going to leak.
Okay, great. Thank you. We got time for one or two more before wrapping things up here. This attendee would like to know how to determine mol as per USP 1207.
So mol is your maximum allowable leakage level, and that's going to be product specific, actually. So there are going to be some drug products that are going to be much more sensitive to oxygen, and so those are going to have a higher or a lower amount of leakage that they can sustain. And so that's really going to be dependent on what your formulation is and what's the likelihood of it, changing over time due to having a bit of a leak in your system. So if you're just worried about microbial contamination, you can probably have a much larger mol. If you are worried about oxygen contamination, then it's going to need to be a lot smaller. So that's kind of where you start with that. But again, it's going to be very specific based on your formulation. The only number that I've seen from FDA, and we've seen it multiple times in letters, is 20 microns. And they're just saying, please use a container closure method with a sensitivity of at least 20. So you can see that all the deterministic methods would meet that.
Okay. We're going to take one more question here. This attendee would like to know, what deterministic do you recommend for PFS? So
the best deterministic test is really going to be whatever you've got access to. So, we have mass extraction at Nelson, so that's going to be my favorite. If you have a method in-house already, definitely that's going to be your favorite. If not, and you're looking to bring one in-house, almost all of them, right? They're going to work in almost all applications. There's just those few caveats that I said would have some special advantages in certain situations. We've definitely done those pre-filled syringes. We've done them in numerous iterations. So inside an auto injector, inside some kind of a safety injector, inside just the naked syringe. We've tested those all successfully with mass extraction. I'm sure that any of the vacuum-based methods would work equally as well. As far as laser headspace, that does require you to have a visible headspace, so some auto injectors wouldn't work with that. And then you would not be able to test them, I don't think, with high voltage leak detection if they were, say, an auto injector, so it was the primary container inside of a secondary container. I think that that method's really restricted to just testing the primary container.
All right. Okay. All right. Well, we are past the top of the hour, so it's going to bring us to the conclusion of today's session. Thank you again, Jennifer, for a really informative, terrific talk. Thank you again to today's sponsor, Nelson Labs. You want to be sure to visit them at nelsonlabs.com. Thank you to everybody out there in the audience for your attendance and participation today. Some of you have asked if the PDF is going to be available for download, and it should be available up there on your screen under the Resources tab. If for some reason it's not there, you're not seeing it, you can follow up, send an email to myself, and I'll make sure that you get it. In addition, this session today will be archived for 1 year on our website, contractpharma.com, and you'll also be receiving an email from us shortly when the session is available for on-demand viewing as well. So thank you again, everyone, for joining us. Have a great rest of your day. Take care.